Hello students, welcome to LEELA'S tutorial. So today's topic is protoplast culture. What do you mean by protoplast? It is a plant cell without cell wall, right? So let us see in detail how you are going to culture it and produce the novel plant varieties, right? So here, this is the structure of plant cell where it consists of rigid cell wall, right? And here you have all the cell components such as nucleus, Golgi apparatus, right, mitochondria, chloroplast, ribosomes, and all the components are enclosed inside the plasma membrane. Okay, so when you remove this cell wall, right, either by enzymatic process or by mechanical process, here the plant cell without cell wall is referred as protoplast. Is that clear? All the components of the plant cell are present, only which is lost is the cell wall. Right? The plant cell without cell wall is referred as protoplast. Okay. So what is the use of this protoplast isolation and protoplast culture when we have many other types of tissue culture such as meristem culture, embryo culture, right? So we have so many cultures, but still why we have to isolate the protoplast? Because here, in order to produce the new varieties or transform the plants by introducing some desired genes of interest, right? So like that, certain applications are there. So first application is in order to produce the genetic transformation. That means you want some plants to get genetically modified. How they get genetically modified? When you introduce certain gene of interest. Now if you want some gene which can be producing increased size of the fruit, or it can be very brightened color of the flower. So like that you can introduce some gene, otherwise you can introduce some gene so that the plant cell will produce certain secondary metabolites. Right? So like that you can transform the protoplast by introducing the gene of interest, right, and then go for the protoplast culture so that that particular plant which is produced by this protoplast culture after genetic modification will produce the plant which will have those beneficial characters, right? And the second application is cell fusion, right? That means the protoplast fusion in order to produce hybrids and hybrids, right? This will discuss in next class in detail. Right? So where we are going to fuse the protoplast of two different plant species. Right? For example, potato and tomato. So you get, when you isolate the protoplast from the tomato plant and the potato plant, what happens? You fuse them with the help of a polyethyl glycol. Right? So here, after fusing, what is obtained? The plantlet which is obtained is called as tomato plant. Right? So in order to get a hybrid or hybrid varieties. And another application is in order to generate unique and novel plants. So already we have the normally existing plants, but you have to improve something so that you get a new variety of plants. Right? It can be in between same species or in between the different plant species. Then the last one is in order to improve the crops. So in case of mango, right? So then you want increased size of the fruit. In case of flowering plants, you want brightened colored flowers, right? So like that you can improve by means of your protoplast modification and culture. Is that clear? So let us see what are the steps involved in this protoplast culture. So the first step, so it consists of 4 different steps. The first step is collection of plant material. Then second step is pre-treatment of the tissue. Third is incubation in enzyme solution. And the fourth one is protoplast purification or protoplast collection. Is that clear? So let us see here. First you are going to take a mature plant which is disease free. Okay, mostly for protoplast culture we take the leaves as explant. So the explant, the plant material is collected and you go for the surface sterilization of the leaf. So surface sterilization can be done by using mercury chloride or sodium hypochlorite, or you can use silver nitrate, right? So any one of the sterilizing agent you can take and go for surface sterilization, so that any dust particles present on it and also any airborne microorganisms can be washed away. Right? So after sterilizing your explant, you are going to take that in an petri plate, and then with the help of scalpel, everything is sterilized. With the help of scalpel, you are going to peel the epidermis. Right? You are going to peel the epidermis, upper and the lower epidermis, gently without rupturing the cells. Right? So without rupturing the cells, you are going to peel off the epidermis of the leaf. So now when you peel off, you may get the cells which are exposed. So now there is no epidermal layer over the leaf, right, or over the leaf fragment. So here now it is a peeled leaf segment where you can see cells with cell wall. Here this is the cell wall. So here also you can see the plasma membrane which is enclosing the plant cell, and you have the cell wall. Right? So now you have to separate these cells and remove the cell wall so that your so that your protoplast will be collected. Right? So what you are going to do, these segments which are peeled off, the epidermal layer is peeled off, they are placed in enzyme solution. Right? So in the enzyme solution, you are going to incubate them from 15 to 30 minutes based on the concentration of the enzyme. Right? So after the incubation, here you can see you get an plasmolized cells and also partially plasmolized cells are obtained. Right? The enzyme which is present in this beaker or the container will digest the cell wall. Right? So as a result, you will get the cell wall debris. Right? Some cells may be damaged, or some cells can be plasmolized and some can be partially plasmolized. So you have to separate your protoplast from all these cell components. Right? So what you do? You go for centrifugation process. So in the centrifugation process, based on the density, you are going to collect the pellet of cells, that is nothing but your protoplast. So all the cell debris, cell wall, everything will be present here, and your protoplast can be collected. The pellet of cells can be collected, and from that you are going to purify the protoplast. That means you are going to see that only protoplast cells are present but not any plant cell, that means with cell wall. Okay. So here you have only the protoplast cells as a clump. Now these clump will be inoculated in plant tissue culture media, right, under suitable artificial environment. Right? So the media is artificially prepared so that the protoplast can survive in that, and also temperature, pH, everything are controlled. Okay. So this clump of cells start dividing to form a colony of cells. Right? The clump of protoplast start dividing to form a colony of cells, then followed by formation of callus. Callus is undifferentiated mass of cells. So now this callus in further incubation, right? So when callus will be formed? When auxin to cytokinin ratio in the media is same. That is, auxin to cytokinin ratio is 1:1. Right? The callus is induced. Once the callus is formed, here you can see the meristemoids are formed. So from each meristemoid, the shoot will be generated. Right? So here the callus differentiation occurs in order to initiate the shoot. Okay. So now these meristemoids will be transferred to the another media or the shooting media, where auxin to cytokinin ratio will be 1:3. Right? So now you will be incubating them so that the shoot formation is initiated and shoots are formed. After the formation of shoot, you will be transferring them to the rooting media, where auxin to cytokinin ratio is 3:1. Right? Auxin must be more so that the roots are initiated. So once the complete organs are formed, that is the shoot and the root system are formed, now you call them as regenerated plantlets. Right? So regenerated plantlets, now they can be transferred to the pots or the soil, or you can give it to the farmers. Right? So here the next step will be hardening process, where the regenerated plants has to get adjusted to the outside environment. Right? So acclimatization, greenhouse, then the plantlets will be shifted to the fields. Okay, so that you can be beneficial with any of the application. Okay, so like that you can produce any desired new variety plants by using your protoplast culture. Okay, in next class we'll discuss about the cell fusion. What are its applications in producing hybrids and hybrid plants?